Hello everyone. Today I will discuss about drug molecule Entecavir. It is an antiviral medication used in the treatment of hepatitis B virus infection. Here is the chemical structure for the molecule. This molecule was discovered by Bristol Myers Squibb pharmaceutical company. The drug molecule got US FDA approval in the year of 2005. I will discuss pilot scale production route of the molecule as described in the OPRD journal in 2018. The synthesis starts with S-carbon as the starting material. They started with 3 kg of this raw material, which was 92% enantiomer pure, and this alpha beta unsaturated carbonyl compound underwent stereoselective epoxidation reaction in presence of hydrogen peroxide and sodium hydroxide. And trifluoroacetic acid helped the protonation of the epoxide ring, followed by nucleophilic attack from this chloride helped the ring opening of that epoxide, protonated epoxide, that provided this chlorohydrin intermediate. Here the secondary alcohol group was protected by using tosyl chloride to get intermediate molecule 4, which was recrystallized using methanol to get 100% enantiomer pure material. Now this ultra pure intermediate reacted with metachloroperbenzoic acid for this exocyclic double bond to be converted into epoxide, and the epoxide ring opening reaction was done by using aqueous sulfuric acid to get this diol intermediate, and this diol was protected by using dimethoxypropane that provided intermediate 7. Now the molecule 7 was treated with sodium methoxide in methanol solvent, and initially it provided cis substituted Favorsky rearrangement product, which one later isomerized to more stable trans isomer. So it is an base mediated Favorsky rearrangement reaction. You know how that Favorsky rearrangement works. Like base helped the deprotonation at the alpha carbon to that carbonyl group. Then the substitution of this halogen, followed by again that nucleophilic attack from this methoxide to that carbonyl, and followed by the that removal of this leaving group. So initially they observed that isomerization process is not completed even after 24 hours, but when MTBE and methanol was used as solvent, then isomerization process completed within shorter time period. Now after getting this ester intermediate, lithium aluminum hydride mediated reduction of this ester group was performed to provide this allylic alcohol, and here the protected diol was again unprotected with the treatment of sulfuric acid, aqueous sulfuric acid, to get diol, and that cleavage of diol was performed by using sodium periodate to give this intermediate 10. And here this oxidation reaction to the alcohol molecule was performed by using hydrogen peroxide in presence of sodium hydroxide. And after column purification, they obtained this molecule with 45% yield at this step. So significant drop of yield happened at this step. Now the intermediate 11, that is an allylic alcohol, here stereoselective epoxidation was performed by using TBHP in presence of vanadium catalyst, and predominantly the beta epoxide was formed, and here both the alcohol groups were protected by using tertiary butyl dimethyl silyl chloride to give intermediate 13. And here very interesting transformation was done. You can see the conversion of epoxide into an allylic alcohol was performed by using lithium TMP reagent in presence of diethyl aluminum chloride. So lithium TMP reagent, they have in situ synthesized that reagent by the reaction of tetramethyl piperidine and N-butyl lithium. So base mediated proton abstraction followed by ring opening leads this transformation to provide this allylic alcohol. Now this allylic alcohol underwent condensation reaction with this protected purine in presence of diode reagent and triphenylphosphine. Diode reagent is diisopropyl azo dicarboxylate. So it is a reaction between this NH and this alcohol, unprotected alcohol, for direct this carbon nitrogen bond formation reaction, and this reaction is called Mitsunobu reaction. So this step leads the formation of intermediate 15, which was treated with aqueous hydrochloric acid to produce the final material. So you can see the role of hydrochloric acid at this final step. The Bog group from this NH group has been removed, as well the silyl group on these both alcohol group also removed. Also you can see this chloride functional group has been converted into the carbonyl here. So at the final step you can see there are multiple role of this aqueous HCl. So the final molecule they have recrystallized using ethanol and water. They have used 95% ethanol and 5% water. So they obtained this molecule 530 gram and with 100% enantiomer pure, and chemical purity was 98%. So you can see they have started this reaction using 3 kg of the starting material and obtained 530 gram of the final product over the 15 steps. So it's overall around coming 9% overall yield. So you can understand how difficult the synthesis. And this drug is sold with different doses like 0.5 milligram dosage usually. 0.5 milligram means half milligram dose. If I remember correctly, that half milligram dose of this drug cost around 80 rupees for a single tablet. So this is a unique process to synthesize the molecule. However, this process might not be suitable for production in even bulkier, larger scale, like ton scale if required to synthesize, then it might be difficult, because fewer steps includes column chromatographic purification. That might be difficult for large scale preparation of the molecule. Thank you.